Ladies and gentlemen, a very warm welcome to the Science Faculty Research Day 2022. Let's welcome the moderator of today's webinar, Professor Liwen Jiang, the Science Faculty Associate Dean for Research. Professor Jiang, please.
Hello, good morning, everyone. My name is Liwen Jiang. On behalf of the Faculty of Science, I would like to welcome you all to the Science Faculty Research Day 2022. So this year's theme is on collaborative and impactful research in science. So to kickstart this research day, may I first invite Professor Chunshan, Dean of Science, for delivering an opening remark. Chunshan, please.
Okay, thank you, Liwen. Good morning, everyone. Welcome to the Faculty Research Day 2022 in the CUHK. Every year, the faculty organize a research day where researchers gather to share ideas and experiences in advancing research and collaboration. We were planning initially to have a in-person meeting in large groups for the research day, but after the soaring fifth wave of COVID-19 pandemic in Hong Kong around the spring, to curb the spread of this pandemic, the Faculty Research Day this year goes virtual again, with the theme that is consistent with our last two research days, that promoting collaborative and impactful research in science. Although we're not meeting physically in large group this year, we hope the Science Faculty Research Day still serve as a good platform to facilitate idea exchange and to encourage collaborative discussion for research across different disciplines. Many of us have been involved in collaboration one with the other. I myself have been involved in developing and leading multi-PI collaboration within one university and university industry collaboration and multi-university collaboration immediately before I joined the CUHK. And I think the advantage of collaboration has been recognized in various areas beyond the science technology. As Mother Teresa said, I can do things you cannot, and you can do things I cannot. Together we can do great things. I think that represents the spirit of a collaboration and the true advantage if we can make use of the collaborative spirit. And of course, in many ways, we're already doing collaboration between any one of you and your student. And that is a small collaboration, but that's within one discipline or within your expertise in general. And we're now talking about collaborative research that crosses discipline beyond your own research comfort zone. And of course, many of us have witnessed the global trend that collaborative research has become more prevalent in science globally, not only because science is a collaborative effort. We stand on the shoulder of a giant, as Isaac Newton said, but also because addressing major scientific and societal problems today require truly multidisciplinary research and development. Therefore, researchers are increasingly required to cross disciplines to be more impactful. But of course, and there are fundamental research that still calls for independent individual research, and that we fully appreciate as well. But recognize the importance of collaborative research. In our faculty, we have identified the 7 strategic research themes. We work with the University Strategic Planning Committee, and we have built our faculty proposed 7 strategic research areas all into the university-wide strategic research themes, and which is now available in the University Strategic Plan. And the emphasis on inter and multidisciplinary research that cross individual unit boundaries in general sense. And we hope this approach will significantly promote multidisciplinary research collaboration and unleash the tremendous potential that our researchers have through the collaborative work. For the Faculty Research Day this year, we're very pleased to have Professor QIN Ling, Choh-Ming Lee Professor of Orthopaedics and Traumatology from CUHK Faculty of Medicine. Several of them know Professor QIN personally, and I have been very pleased to have the pleasure and honor to actually know Professor QIN through our several meetings together. Professor QIN has been leading and with a demonstrated track record of highly successful and impactful collaborative research, including his leadership to develop a competitive research grant, such as a CRG, GRG, CRF, TRS, and AOE. And he will share his insight today with us on the impactful and translational research, and that will involve collaboration with scientists from diverse fields. Professor Ching is also the director of CUHK Hong Kong Shenzhen Innovation Technology Research Institute, and that's located in Futian, and so it's also commonly referred to as Futian Research Institute in the Lomazhou area. His sharing and insight on this aspect will also be very useful for research in the Faculty of Science. As a related effort, our science faculty members in physics, chemistry, and life science are actually involved in developing new collaborative enterprise in the CUHK Great Bay University Joint Institute of Advanced Materials and Green Energy Research in Dongguan, and which Dongguan city government have pledged major funding support for this new joint institute. An MOU has been signed by both universities recently. And the faculty has started a new seed grant in 2020-2021 for promoting collaborative research across the disciplinary boundaries in the faculty, with the support initially using the fund from CUHK Project Impact Fund. And we're now actually making that a standard practice with our faculty budget to continuously promote the collaborative seed grant program, which is called the Faculty of Science Collaborative Research Impact Matching Scheme, CRIMS. The scheme has been supporting a number of faculty research teams since 2021. In this connection, and we feature 5 of the collaborative research team funded by the CRIMS research scheme. And today we have 5 research teams represented by the team leaders, Professor Manning Chen in Earth System Science, Professor Michael Kenneth Chen in Life Science, Professor Xinhui Lu in Physics, and Professor Yingying Wei in Statistics, and Professor Hongfeng Yang in Earth System Science. They will share the highlight and their experience and perspective based on their latest collaborative research project supported by the CRIMS 2020-2021. As indicated in a quote from Henry Ford, and the father of modern automotive industry or the car manufacturing industry, coming together is a beginning, staying together is a progress, and working together is success. Over the last 10 years, the faculty has successfully secured 22 collaborative research grants, that's a CRF project, and 3 area of excellence projects funded by RGC. Riding on the excellent track record of the collaborative research, we continue to strive promoting and advancing collaborative research across the disciplines in the faculty. It is our hope that the faculty research day-to-day will foster the faculty initiative to promote the discussion toward collaborative research, strengthening existing collaboration, more importantly, facilitate development of new collaborative research between researchers in different disciplines, and particularly involving our younger faculty members and junior faculty members who recently joined the faculty. We hope this will provide you with an overview of how the faculty support collaborative research and how these collaborative researchers have been successful riding on this seed grant program for collaboration. So I look forward to a stimulating discussion with the research day-to-day and with active participation and lively discussion. Thank you.
Thank you, Professor Song and Professor Jiang. May I now invite the speakers and panelists to switch on your video cam to take virtual group photos together. Professor, please. Okay, we've got the photos taken. So may I now invite Professor Jiang to introduce our keynote speaker, Professor QIN Ling. Professor Jiang, please.
All right, good morning again. It's a great honor for me to introduce today's keynote speaker, Professor QIN Ling. Is well known. I don't need to introduce too much. Okay, just want to say a few things. So Professor QIN Ling is a Choh-Ming professor at CUHK. He's the director of the CUHK Hong Kong-Shenzhen Innovation and Technology Research Institute, and he has published over 360 papers, okay, over the years, and he has received over 30 major grants. You name it, all the grants that are available in Hong Kong, including AOE, TLS, CLF, okay, all the grants, okay. And also, I think his work has been recently highlighted by Nature and Science, okay. So please join me to welcome Professor QIN Ling.
Thank you, Professor Jiang and Professor Song for your invitation. So it's now full screen or whether display is okay?
Yes, yes.
Not this screen, okay, okay.
I think you need to change the display setting.
Yes, right, okay.
Yeah.
Oh, sorry.
And then you click the play button on the bottom right. Yeah. And this and swap the, yeah, that's it. Thanks.
Okay, thank you. Okay, thank you again for the kind invitation. So the title is somehow given. It's a really challenging big one, but Professor Song already addressed how important is the collaboration. I also show here, but the major purpose is to start some collaborations because you already mentioned in the past years, I got several grants, including consecutively for GIF over the past 6 years. But as mentioned, I'm still looking for collaborations with our faculty members and students. We have some existing collaboration already, so we can further enhance. So if you look at the website, there are many features about the importance of collaborations. Of course, as you see here, collaborations increases research impact, and always say good science is never conducted in vacuum. Scientific collaboration is a large part of successful research, and without collaboration, scientists can miss out good ideas. I benefited from such collaborations and ideas, of course. We know travel is important for collaboration, and we have good examples in our field, musculoskeletal research. We indeed start intensive collaboration with world leading institutions in relevant field, and launched MRN, musculoskeletal regeneration research network with top institutions consecutively over the past years, but discontinued right, since 2019 and due to no reasons. So we try to overcome this. At the moment, just like today, we through collaboration through Zoom, right? Okay, to share with you, I try to address few points. Form of research collaborations may not inclusive. Impact and high impact research, mainly with regards to medical science here, and personal engagement towards AOE, as Professor Jiang mentioned. And then last, if still time, on the recent development in Greater Bay Area here, focus on the Futian Institute. So we all know academic aspects. We have joint grant application, joint publication, education, research programs, joint R&D. And nowadays, of course, we have the mission also collaborate with industrial downstream part, and including knowledge transfer, joint R&D, particularly to support the industrial part. And we also conduct the contract studies or provide service. So academically, we know the impact of research collaboration on the productivity. There are publications over 400 academic scientists, particularly here, the data from USA, demonstrating that such collaboration is very effective for population productivity. We also have similar experience. And I also learned that the coming QS ranking will also focus on international collaborations to see the multi-authorship with different institutions internationally. So from the industrial aspects, we know that research and innovation policy aims to boost research output and university industrial collaborations, in part to allow firms to access leading scientific knowledge. And we also know that apart from their missions, university in many countries now expect to contribute to innovation in the relevant regions. This is a published work in Nature a few years back, the University of Future will be inclusive of broad swatch of the population actively engaged in issues that concern them, relatively open to commercial influence, and fundamentally interdisciplinary, that means collaboration in its approach in both teaching and research. So this is a conventional model, right? That university contribute to science publications, and through our graduate students, they may join industrial. So industrial may get that contribute to the society separately, but the interplay is our product, our students. Now, we have to be more effective, and the new demands, as mentioned before, university industrial partnership is so important, so we can join hands to conduct the joint research, hiring, and exchange the technical licensing directly. Our university is very proactive nowadays in this area. So concerning, of course, the impact, we try to create impact, and not only for publication, but also for research grant, mainly also its downstream part for its application. We all are familiar with the research excellence framework a few years back, the research assessment exercise. Now, impact is any effort on change or benefit to the economy, society, culture, public policy, or services, health, the environment, or quality of life beyond academia. So this is a new demands. So this requires, of course, our strategy towards high impact research. Now, concerning the spectrum of impacts, I here refer mainly to the medical side. We have scientific impact, right? Improve our understanding, open new research directions, mainly based on the citation somehow. And the socioeconomic impact, we look at the technical development, invention, new implementation by using the index patents, products, standards, regulations. And also clinical impact, our practice health information sharing. So as fundamental issue, of course, 3 I's is important towards high impact. That means innovation, interdisciplinary. So this is also the aim of today's research day. And finally, integration is important, particularly relevant for our medical sites. And you are also aware of that this year's exercise in Hong Kong, 80 universities, right? Based on the data, we know 70% rated internationally excellent, among them 25 world leading, which is really excellent. So commented by Charles Tong that the university performed well in research impact, which reveals that the universities did a great job in transferring their research into innovative solutions, bring substantial benefits to society and significant tangible changes to people's life. So all fit in our strategy. Though this group-based, not individual-based, but we can name a few from our faculty. Professor Dennis Lowe, we know his innovation application through the non-invasive prenatal testing, right, technology. And in engineering faculty, we know recently Sang Tang is already stock listed, which have a really huge impact through AI technology, right? So all behind is science, technology, intensive collaboration. In fact, in our area, medical products particularly, so level of collaborations from STI, FDA to IPO. So we know for project, probably for publication, we meet 10 people around. So if we go for R&D, more people involved. If you want to go to IPO, particularly as I mentioned, the medical products, it's really huge, people involved. So collaboration definitely is important. I also learned that among audience, we also have students, younger faculties. So somehow we also look at the report from, for example, final report of Task Force on High Impact Research at American University. I just quoted here. Lots of people want to do high impact research while still a graduate student to postdoc or postdoc. That's great if it's possible, but it's even more important to first establish your career independently or by joining a good group, emphasizing on collaborations. In the current academic system, it is very important to develop a strong publication record early. It is also important to get great training. So seek out a top research group who will mentor you, even if it is not an area you would ideally work long-term, right? But just make efforts to collaborate. So what are the high impact research topics in general? So if our career becomes more established, we will get more and more freedom to choose the topics you want to work on. Personal fit is especially important in research because the most productive researchers have far more output than the media. We should look for area for long-term sustainability. I think all of us are working on that, right? So, and for a field where you have a realistic chance, right, being in the top 10%, if not top 1%, it's really challenging nowadays to get academic position in top universities, even in regular universities. So based on those, that also I have personal, understanding to a certain extent. So I would like to share some engagement towards AOE in my field. So I will not go too much into details, but in general view. So I'm leading an innovative orthopedic biomaterial and drug translational research group in Faculty of Medicine. And this is a few products we are working on. Especially, I would like to emphasize the very attractive biodegradable metals, mechanism-based implants for our application. But if you see here, we need to identify indication, clinical indication first, where we need it at all. And the second, since we have to put into the body, so preclinical testing is substantially important before getting to clinical trials. Of course, working on industrial design products and then regulatory body approval is the final stage before launching in the market. And of course, the first generation is there and their feedback, we have to improve further. So what is magnesium? We all know periodic table, right? Magnesium is next to the calcium. Calcium, probably we all know about that. Daily nutrition supplementation or consumption contains calcium. So interesting that magnesium is also a very important element, although proportionally to calcium is low. So magnesium ions existing and known as over 300 chemical reactions in our body in almost all systems in our body, particularly in nerve and musculoskeletal. So another attraction for our orthopedics is that we can use the mechanism to make implants. And if you compare its mechanical properties, it's between very hard, stiff titanium and stainless steel. That is conventional implants we are using nowadays. And compare with the cortical bone and also in chemistry polymer, right? So in between. Mechanical property is very attractive. And the resources is huge, biodegradable. Theoretically, it can be used as implant for avoiding second operation, surgical operation, very attractive, particularly for me. This is my own photo. It's early 90s. I participated in World Championship of snow bike. I got injury, a cheekbone fracture, double fracture. So immediately, operated after, getting down the swollen. And then no problem. A year later, doctor mentioned me, you should make another appointment for removal. Then I had a huge psychological burden, really. If someone injured, no worry, they would seek for operation as soon as possible. But if it is healed, really it's a big burden, apart from economic ones, right? So I then, after joining CUHK, I had, through collaboration, travel, and we built up, a team to look at biodegradable metals. Immediately, I recall my cases. So I think it must be attractive. And that was 2010 in Shanghai with metallic scientists, engineers, and also doctors. And a year later, here with the company here registered in Hong Kong, but the company is based in Dongguan, working on magnesium. We established university industrial collaborations in R&D of biodegradable magnesium for medical application. So if we look at the data, we're available, basically many engineers working on that, but none has reached clinical application. So the major burden is that mentioned, indication, pre-testing, regulatory requirement, and so on and so forth. Indeed, if as implants for human body, it's a class 3 medical device, you need a lot of steps beyond science. Okay, regulatory issue. One thing is about safety assessment. And we understand, basically, there's no international ISO standard for testing. So we formed the international group to look at the clinical applications by establishing ISO new standards. And this is the group we formed and joined Think and Tank workshop in FDA in 10 years ago. And then go through all these efforts. And when we then work on China FDA, and now 2 years ago, finally got the standards approved. So there were existing standards for testing, as I mentioned. All this would unfavorable for our application because they mainly concern the in vitro testing, not the human body. So this is a new one launched. With our contribution, we did a lot of research to confirm that there difference between in vitro and in vivo situation. If that one used for in vivo, we should not have such cell toxicity concerns. So with the title Recommendation for Modifying Current Cell Cytotoxicity Test Standards for Biodegradable Magnesium-Based Materials as a mandatory paper. And then we are able to conduct first a clinical study in China for femoral head necrosis. This is a magnesium screw after implantation to fix the bony flap, and then we see at the baseline, and 12 months later it's degraded with time. And compare with the conventional one without screw, we see with screw it's degraded, and its degradation enhance the bony flap bone mineral density, which means make the implanted bone vital. And this is a particularly interesting case, a young man falling down with high energy femoral neck fracture, and conventionally use the metal screw, permanent metal screw. But unfortunately, 26 months later, got osteonecrosis. Normally, surgeons recommend for hip replacement. And then we say we have new technology you would try, and he accepted without any costs involved. So we remove the permanent metal screws and using the bony flap magnesium screws. You will see here magnesium screws and bone mineral density around. 2 years later, showing significant improvement. This is the boy. After that, he can work. And if we use the conventional hip replacement for younger age, he probably need to do 2 more operations for replacement because the implant have lifespan and he is so young. So it's very exciting. And finally, with corrective efforts, we are able to get CFDA approval for starting clinical trials for product registration in 2019. And similarly, we also know we have set up in Shenzhen Academy Sciences, and we built up a translational center to make use of such platforms. We are also able to produce 3D printed materials and by forming the startup. And finally, we got the multi-center clinical trials last year and with PI from Jesuitan Hospital. Now we have 6 hospitals studied with our product multi-center testing. And we hope end of next year, we should have the product available in the market. And our collective efforts have been also summarized in a review paper in Materials Today with our collaborative work here. So innovation is important as a driving force. Okay, so if you see here in orthopedics, the driving force behind is the rising technology advancement in this, including our biodegradable materials. And we also understand surgeon geriatric population. Now the population is aging. Their regenerative potential is poor. So we need to work on innovative materials with bioactivity to enhance fractures healing. So traditionally with the permanent metal, as you can see here, there are different approaches. And we tested biodegradable metal compared with the polymer. Nowadays, polymer is quite inert. After 2 years, even no degradation. Used to be designed for degradation, but too fast. And you see here biodegradable magnesium and stimulate a lot of bone formation reported by others. And this is our own data. See magnesium implant here, and you see here many bone formation using labeling. You show those are new bones. We published such phenomenon. And interestingly, we look at, instead of next to the magnesium degraded parts, the bone in the peripheral is active. So what are they? They are periosteals, the membrane in the bone. So after removal of the membrane, the bone membrane, no bone formation. But if one piece left, you see a dramatic bone formation. So what are in the bone membranes? Periosteals. There are a lot of nerve endings. There are a lot of periosteum-derived stem cells. We know stem cell is able to generate a new bone. So in fact, we explain such a situation, but if the degradation is uncontrollable, we may have some challenge for really application. So that's why after extensive review, understanding the healing feature and degradation properties, we are developing now hybrid system. This is the proof concept study using conventional intramedullary nail for fixation, where we incorporated the magnesium pin by drilling the hole so that they can degrade over time without sacrificing the mechanical strength of the fixation. And we demonstrated its gradual release. Indeed, in the magnesium group, we see a lot of callus formation, particularly in the early phase, which is important for fracture region without affecting later on the bone remodeling to get normal. Mechanically, there are around 30% improvement. So the underlying mechanism is related to the nerve ending free nerve ending, sensory nerve ending, where we see a lot of CGRP, neuron growth factor proteins, highly expressed in magnesium group. And where are those neuron growth factor CGRP formed? Basically, it's in the root dorsal ganglion, and we see in the magnesium group the high upregulated synthesis of the CGRP. And then in vitro, we can confirm the release to the nerve ending. So with all this, we have 3 new innovations or novelties. One is how the magnesium ion stimulates the sensory nerve to release CGRP. And the second, the CGRP on the osteogenic differentiation of periosome-derived stem cells and related underlying molecular mechanisms. And finally, the innovation of the design. Okay, with that, it's been published in Nature Medicine with research highlights by Nature Review Endocrinology, emphasizing on the neuron origin of the osteogenic effects of magnesium. So if you see the author list, we have collaboration from Germany and from Beijing, Mainland, and Hong Kong. And now we're moving forwards to solve clinical problems, particularly in atypical fracture for elderly people who take drugs for prevention of osteoporosis. But if they get fracture and repair, then it's poor. So we get the patent from US and the PRC. And our underlying preclinical study demonstrated that magnesium facilitates the healing of atypical femoral fracture using a single cell transcriptomic study. I think we have experts here, so we can collaborate. Important is, again, collaboration from Sweden, who provide clinical samples for us. We do not have sufficient here. And Professor Regis O'Keefe from WashU, US, and of course, also our group. And such important finding has clinical meaning, so been highlighted by Science recently. So also we are very pleased our collective efforts has been featured in Nature a few years back. So with that, of course, we look at the innovation on developing bioactive implant for fracture fixation, especially for aging population. So I just go quickly. So we have exhibition to show our innovation and reported also in the public that receive really good feedback that stimulate merge our collective efforts to realize our product for clinical application. So all these are foundations for our efforts towards AOE. So I just summarize a few key issues for the AOE. I appreciate Professor Jiang Liwen. You were in the selection panel internally and gave very good feedback for us to improve our presentations. So of course, clinical background is important, aging population, and the more fractures we see. And of course, the demands, costs are high. Those are standard for us to justify our applications for long-term sustainable development. And of course, our understanding on the underlying mechanisms on the impairment of bone regeneration. I will not go to details, just to show. And then this is one. Another is osteonecrosis, which can be result from trauma, as mentioned before, the drug use, and eventually alcohol drink. Right, all this can induce such problems. The repair is challenge. So also one sports injury on the ACL ligament issue, and Professor Young's our department head is working in this area. So we summarize all these challenges. The bone quality become poor, MSC quality become poor, and the quantity as well. So all those are common features. So we looking for certain bioactive implants for surgery. So that's why we look for these, we introduce the magnesium, why it is important. and already mentioned, it's biodegradable. And we form the group with industrial being able to have high purity that meet our clinical application demands. And innovatively, we also have collaborated from CTU, Professor Lu, and who published in Nature and other relevant papers on how to advance alloying materials and surface modification for the implants, which also important for us. And of course, mentioned our innovation and also its biodegradable products. Apart from magnesium ions, it's create alkaline environment and hydrogen gas formation, which have been proven scientifically being able to anti-infections and also other features. So all these are good ones. So we form a team collaboration again. So from basic translational research aspects from various universities, from material science and medical device aspects, and finally, clinical research. So we just justified that we got a significant number of GIF grants, collaborative research fund. So move to scene-based and now to a stage to become area of excellence. So important track records in the past, and just to show the incomplete data. Those are yellow one numbers in the proposal, right? Publication, we committed to 15, and for scene-based, we already reached 36, even before completion of the study. And book chapters, conference objects, patents, right? We have new patents and also awards. All these are really not mentioned in scene-based, but we achieved. So with some, we summarize that in the group, we are able to demonstrate our scientific potential by using the past track records in those top journals and monographs, which is important to show that we are able to manage, lead in the field development, innovation as an index, those patents, both in PRC and the US. And international peer recognition in group and in person as well. So finally, we say mission and goals. I'm not going to show the detail, the proposed what going to do, but just you show the background and our approach. So from basic research, we try to do is understand the byproducts, the degradation products, scientifically and so on and so forth. Okay. Clinical trial product registration, which we have to go from upstream, midstream to downstream part. At the same time, as an education institution, we try to build up multidisciplinary collaborative education platforms from biology, engineering, and medicine, clinical medicine as well. So of course, in order to manage a project, we have to build up the management mechanism, the teams, where I served as a principal coordinator for those basic and translational R&D material device and clinical trial studies. And we know we have still weakness, so we invited advisory people from overseas and domestic ones to join our advisory board from, as mentioned, academia areas, in R&D centers, and in industrial sectors. And probably we just know that recently we also show our hybrid system and Geneva Convention, and we got the gold medal. And this also encouraged us to move forwards quickly. In fact, through all the years for Class Medical 3 devices, we know the 正常学研医用 means regulatory policy, industrial, academia, R&D, medical side, and uses. Our patients are the key elements of success, also the foundation for our AOE. So if that we can link up together efficiently, we can achieve our ultimate goal more faster. So with that, I think we need to explore further, need a further downstream partners, and our CUHK's development in Great Bay Area may serve such purpose. So I'm quite honored to be appointed as a director and a legal person for Futian Innovation Technology Research Institute. So I just use 5 minutes, right, to introduce that. The slides are in Chinese. I just go quickly. So we know our university making great efforts starting from 2006. We collaborating with CET in Shenzhen with Chinese Academy of Sciences. And then our own research institute in 2011. And then 14, the Shenzhen site, CUHK. And then Zhongchuang Enterprise Entrepreneurship Innovation Hub in CUI. And 2 years ago, we started Futian Institute. Now Professor Song leading another initiative in Dongguan, right? So, and that should be at the end. And the purpose, we all know, we try to convert our innovation findings, our staffs, students, really the innovation for application. So we make use of this platform and develop in Greater Bay Area for the overall theme, of course, that serves the overall national development. Okay, at the moment we have one project there on the intelligent robotic programs. And probably we all know where the Futian Institute, just across the board. This is the area to be on the way for as the International Innovation Center in Greater Bay Area. And this is the building where we are in Futian Baoshui Qu, right? And a newly decorated exhibition hall. The information dissemination by showing as a showcase for our R&D is very important to attract the collaboration locally, regionally, and internationally. So of course, we have great opportunity for our innovation, right? All this we all know, and the central government supports this region to become international innovation center, which is policy-wise is very important. And last September, both Shenzhen, Numerical City, and our Hong Kong signed agreement to collaborate, so-called 一区两园, right? And a month later, we formally assigned our, agreement, strategy development contract with the Futian government. So, as mentioned, because of COVID, we really could not realize as we initially designed, but we are making efforts to prepare for. And we know these are 6 important areas for development. The medical technology, new materials, big data, AI, robotics, the nano electronics, and the fintech. Okay, all this are the one. Now, if you are interested, of course, you are also most welcome to join this. And we have one project already launched, held by Professor Liu Yinghui, and second one in preparation, as basically is approved, but need to start, and by our dean, Francis Chen, and other members indicate their interest, and some of them already some proposal. So individual efforts is important, but if we can make use of all these strengths, we really can develop nicely and start collaborations, make it development more efficient. So we are also constructing our website for facilitate really good collaborations with the slogan innovation and transformation, smart manufacturing in Greater Bay Area. So to summarize, I think as you may or I have to appreciate is that all these achievements are attributed by many parties, including our faculty members, students, our industrial partners, eventually regulatory bodies as well. So collaboration is an essential strategy towards high impact research. We hope so. And innovation and R&D is definitely the key. And research facilities, funding support, teaming, and management are also elements underlying the collaborations. With that, I would like to thank Professor Song and Professor Zhang Liwen's invitation again for this nice research event, and this will definitely also facilitate further collaborations between faculties or among faculties. Thank you.
All right, thank you very much, Professor QIN Ling. Well, wonderful sharing. Okay, I'm sure there will be many questions. Okay, now we open the floor for questions. You can just unmute yourself and ask the question directly. Questions?
Professor Qian, first of all, I really want to thank you and from the bottom of our heart in that your presentation has been truly inspiring, and your research has been so impressive. Through the collaborative work, you have developed a new way of the bone fracture repair, and much better than the traditional way of the so-called steel needle or iron steel needle. And now you no longer need that and no longer need to do the second surgery to take it out. But more important, I think the collaboration and also in-depth research shown that it actually even stimulates the neuronal development such that it facilitates the structure become part of the integral body of the bone structure. And that is really wonderful. I think the way that you have reached out with the industry and the government, as you said, through the regulatory aspect, and you work with them, actively work with them, and you actually create a new regulation. And that become a new standard for the field of practice. And that's very remarkable and truly impressive. So thank you very much. I'd just like to say, ask a question that, and for the benefit of our young faculty members who are relatively new, who are starting as independent researchers, how do you reach out to build the teams in an early stage? If you could share that, it would be very helpful to our junior faculty members.
Okay, thank you, Professor Song, for summarizing what I just mentioned. Yes, the main driving force for scientific research is from our team here in Hong Kong and partially also across the board in Seattle, in Shenzhen, where I have a translational medical center. They have staff for long-term development. Here we have students who may stay with us for a period of time, and they are keen, of course, mainly on scientific output. So if they demonstrate well, of course, we are able to maintain them and eventually send out for overseas, and university have new STEM professorship, including the junior one, if they can, demonstrate further with overseas experience as well. So we are making efforts to promote that, and instead of just assign duties to them, normally we try to initiate a discussion. No questions due. Let them to become more proactive. Unlike Westerners, normally we see the Westerners, they are more proactive to raise questions, discuss here. We here normally keep quiet. So I think we have to change this environment to stimulate them to, think from themselves. And if they have really good ideas, we should incorporate into the overall development. This will help them to become more and more independent. But we also need to cultivate them that such efforts should be collectively done with good supervision, mentorship from our side, because we introduce a lot of partners for them, right? Our friends, whatever partners, they give free service to us in favor of our training and the students, right? They benefit also from that. We need to do both independently and collaboratively. So help them to build up this. And in fact, the last paper I mentioned with comments from science are from our junior faculties and postdocs, our first author, and rated internally here get the best impact paper award from our faculty as well. So this will help them, right? So for our supervisor, I think we should become more open, right? Let them do and tell them not the tricks, but the basic principles, and give them freedom. And this will also benefit, because we are so busy nowadays, are not able to read so many. We have to, rely on our students to read the paper for us. So as you do as well, we organize in journal club. And of course, I also search the relevant ones and let them to do journal club, and why they are good, where they still have limitation, where we still have the niche we can do better than them. So all this will help to stimulate their interests and let them become more proactive. And in the future, they should become more independent easily. If we hope that we hope to keep those, but we have limitations to keep all of them, so that they can really find a good job in the future as our collaborators. So we can really expand our resources, just like to develop in Great Bay areas. Thank you.
Yeah, thank you. Appreciate it. Yes.
All right, Professor Qin Ling, I have two small questions about the institute in Futian. So I understand many of the current applications mainly from engineering or medicines. Do you see any potential chances for the future enrollment from the faculty of science? That's one. The second one is that I know everyone is quite busy, right? So what would be the challenge for someone like us to build another big project in the Futian?
Yes, right. Really good questions. The opportunity for our faculty to join the Futian Institute. Basically, it's open, as Professor Song knows, right? So all are part of that institute. Now, the only that you have more opportunities and to develop in Dongguan, so probably the internal mobilization could be diluted a little bit. Okay, but nevertheless, we do have similar projects, and we collaborating with our Faculty of Sciences, join the population, join the CIF scene-based AOE, all there. For Futian as well, in my proposal, we have also faculty from science to become part of it. Now, what I haven't mentioned is that I'm working on block funding opportunity with Futian instead of individual approach, because I see the limitations. If we have individual approach, then the leader, if somehow retired, and the whole group may face big challenge, And if we have many projects and start and we group them together with defined focus R&D areas, so everybody can take care of each other, collaborate with each other, would not so much affected by the individual leader in the programs. So we have the whole proposal, initial proposal, and then with our VC, hopefully that soon after COVID free travel, we can really send a delegation there to talk with the Futian to understand the pros and the cons of individual versus group approach. So most welcome if you really would like to join and submit proposal individually. So we have to do both individual first and then group at the same time to see. I wish that we could get the block funding to support our development. This will make our administration much more easier. So far, the major challenge facing now in Futian is administration side, the lack of funding. You know? Although individual of us have many funding, but we can't not do much. The second question, sorry, can you remind me again?
Everyone's quite busy. You want to build a new project.
Oh, yeah, right.
So with the challenge, yeah.
Yes. Well, I think one cases of resources is important to start with, and then if we compare with CUHK, right? Although our CUHK case law is declining compared with the very beginning, okay, but there were some of us still keep the centers. So for me, when I first joined there, of course, supported by university and faculty and the department. So I was taught to build up a center with 40 people. So I could not imagine, right? Because it's already very hard for me to keep a small group for 10 people, right? I have to work very hard and to get a funding, you know, all soft money, basically. And then now we have over 50 staff there already because the manpower, the academic leaders are important. We started with our staff moving over there, you know, graduates moving there. Now my postdoc that time with me become vice director of research division, right, and the vice director of the institute, higher than me because I'm still center director. They are higher me. Okay, even my newly recruited staff all moving up. So that's good because they can, you know, develop by themselves under our general, you know, supervision, and we give them wisdom, create possibilities for collaborations locally and internationally. And indeed, they got a lot of opportunity to collaborate with my overseas partners, right? Got a lot of funding from the most, and so on and so forth. And that would not be possible for us from Hong Kong to apply for such grants. So, you know, to identify good people to start there with essential support is important, long-term sustainable support. And with the time, they can create by themselves. So this will be much easier. So I said probably I can just play as an advisor instead of center director. But somehow they say, still insist that I still keep the position of center director because that can attract more people to come to join the center. I hope this would be a similar situation in Futian, but I know the challenge is much, much bigger because all the administration from CUHK is done by the institution there. Okay, also our staff there. But in Futian, I have to take care by myself. This is a challenge. So it depends on in which scenario. So, but worthwhile because Futian is our own, you know, R&D centers. We indeed need more efforts to develop. Thank you.
Thank you. I see Xiaodan has a question.
Yes. Can you hear me?
Yeah.
Perfect. Thank you for the sharing. It's very wonderful innovation. So I just have a question about your new technology. So you use MG as the metal nail. So will it cause because it will be absorbed by the body, right? So will it cause overdose problem for MG side effect?
Side effects. Basically,
because there will be overdose problem, right?
Basically, we're talking about the metal. Okay, also ions can be ions. Concerning the metal, the degradation is important. If it is too fast, the local concentration of magnesium, pH value, as well as the hydrogen gas production could be more. So this would harm the local tissue, right? So that's why the purity of magnesium is important. Now we are able to produce from initial 2N 90%, which seems to be high, but really as a pure material is not. Now we are able to get to 4N, 99.99% purity. That helps to reduce the degradation and make the metal more durable to meet our requirement. But sometimes pure magnesium is not hard enough for certain applications. So engineers work on the alloying technique with other materials, techniques together with zinc, with calcium. Indeed, we tested all this and selected the one we think is more favorable for designing application with the specification for areas where they're weight bearing, load bearing, or less, just for temporary fixation. So all depends on. So concerning adverse effects, I said if too much, it might, but for regulatory body, they mainly concerning the systemic adverse effects. So then we have to understand the degradation itself and its secretion. After degradation, part of gas, hydrogen gas that dissolve very fast, sometimes good for certain disease, just like osteonecrosis. And hydrogen, the local elevation of the pH value, which is really good to anti-infections in favor of osteogenesis, bone formation, that has been demonstrated, and also in our studies. Mainly the ions. So basically, the kidney is the major organ for balancing the magnesium ions. So far, even with clinical studies, we haven't found any challenges on the change of zero magnesium level. All these are within the normal range. So far, systemic concern has not been reported. And the local then is not regulatory bodies concern. It's on the efficacy. So that's why we have to balance to look at the overall efficacy, whether after implementation it's getting better. We did publish a lot animal model studies and also demonstrated in clinical studies at the moment, both for hip replacement, for necrosis repairment, and also for bone defect repairment.
Thank you.
Thank you.
So actually, I have another question about your SIAT. The current policy for CUHK is you only have 2 months time release to handle the project in Shenzhen. Right? So how can you be how can a PI in the Hong Kong starting campus to be a leader in the SIAT project?
Oh, actually, we have the policy internally. Every month we have 10 days, right? Can work on any affiliation to our CUHK, including the Dongguan one as well. I think the same policy will apply. Okay, so by averaging what I did in the past, basically every month I only spend 4 or 5 days over there. Now, of course, over the past 2 or more years, stayed in Hong Kong. But we work in office, we can give around Zoom meetings, give instructions, and for joint publication, we can spend our time. That, serve the purpose for both CHK because all the physicians there. So this is also now the new evaluation criteria for the QS ranking, looking at the multi-affiliation to encourage collaborations. So far, I think we do not see any challenges on this the internal policy allow us for 10 days per month.
Okay, thank you.
All right, because of time, I think we could entertain one last question, if there's any. Okay, if not,
Hello.
tell me. No?
Hello.
We have one question from the audience, and
Okay,
I just
one
unmute him.
last question. Okay.
Hello, everyone. Thank you very much, Professor Ling, for your presentation. It was very interesting for me. I have one question about the magnesium. How do you know that it is not react with other compounds in the body, like hydrogen peroxide or other metals in the blood or other things? Because it can react possibly with a lot of compounds, and it can affect on the degradation or produce some toxic materials during the treatment time. So that's a question mark. Thank you.
Okay, again, it's on the diversity facts, how we could know. Normally, as I mentioned, for regulatory body, they only check the magnesium ions, or if other alloying materials are in the serum. So we take the blood. Basically, we understand its degradation behavior, and in our body is not linear. They degrade first fast, rather fast, later on plateau, because on the surface there are also other the mineral forms that prevent somehow further degradation. So we get a sensitive time for testing. Normally just 1 day, within 1 week, within 2 weeks time, we get the serum. Okay, every 3 days for testing. The later one never show the the higher serum level. But in spite of this, all, as I mentioned, secreted by the kidney in practice, and theoretically we can estimate, in fact, how much degraded over the time, and how much if that into the serum without the secretion by kidney, how many percent can be increased. Okay, basically no such concerns. For example, for the largest screw for hip, just like a small finger, say, it degraded totally within 3 years time, a little bit faster than in the early phase, as mentioned. So this are controlled by the regulatory body. But the local one, as mentioned, they did not check. They only say the efficacy. If the efficacy is good, okay, it's not their concern. Their concern only the systemic ones. Thank you.
All right, so I think because of time, we need to move on to the next talk. So please join me to thank Professor Chen Ling again for a wonderful talk, and also a lot about sharing discussion. Thank you again.
Thank you.
Thank you. Thank you.
Yeah.
Okay.
All right, may we now invite Professor KB Wong, the Director of School of Life Sciences, to introduce our next speaker, Professor Michael Kenneth Chang, please.
Okay, thank you. Okay, it's actually my pleasure to introduce the next speaker, Professor Michael Chen. Michael Chen's research actually lies at the interface between biology and chemistry. Professor Chen obtained his bachelor degree in chemistry from Harvard College, and he got his PhD in also chemistry in UC Berkeley, where he worked on the synthesis of about inorganic model compounds of manganese oxygen evolving complex of photosystem 2. And then he did his postdoc training with Professor Doug Reed at the CUHK, where he learned to use the protein crystallography to determine the structure of metalloproteins. And Michael began his independent research at the Ohio State University, where he continued his interest in elucidating the structures of metal enzymes and other proteins. He's arguably best known for his co-discovery of the 22nd genetically coded amino acid pyrrolysine, from the structure of the methylamine methyltransferase. And this discovery led him to undertake research focused on using the pyrrolysine system to incorporate unnatural amino acids into proteins, which remains one of his research focus today. And then after joining us at the CUHK, his group initiated a novel research area, which is will be the focus of his talk today, on the using of natural protein crystal produced by a bacterium, bacillus. And then his group has demonstrated that these crystals can be used as a platform for the delivery of protein drugs, and as well as a novel scaffold for direct immobilizations of enzyme for biocatalysts. And without further ado, let's welcome Michael to present his work on this topic.
Thank you, KB, for the kind introduction, and thank you for the faculty for inviting me to present. Can you hear me? I just want to check before.
Yes, we can hear you.
Start. Okay. Okay. So enzymes are actually very commonly are important components in industrial catalysis.
Excuse me, Professor Che, we can see your PowerPoint sharing.
Okay. Sorry. Let me make sure that's working. That's why it didn't work. How about now? Can you see it?
Yes, I can see it now.
Okay, good. Let me put the chat over here. Uh-oh. Okay, why isn't it? Okay. Okay, I can do it this way. Okay, so enzymes are important for industrial catalysis. They're found in detergents, they're used for food processing, they're used to make commodity chemicals, and they're also important in making chiral drugs that are used to treat human health. Okay, so when you're in industrial catalysis, a very common thing that people do is to immobilize the enzyme. They can cross-link the enzyme like this. They can covalently bind the enzyme to some support. You can entrap the enzyme. You can encapsulate the enzyme in some type of bilayer, or you can absorb it to a support of some type. But the problem with all of these approaches is that they're expensive. And the reason for this is that for traditional immobilization technologies, what you have to do is express the protein, you sonicate the cell, you purify the protein, and you concentrate it. Then you have to combine it with some technology to immobilize it and couple it, and then you wash it, and then you have your final immobilized enzyme. If you could do all of this in one single step, you basically directly immobilize the enzyme in bacteria, do an autolysis and wash, you save a lot of money. It would be much cheaper and more efficient to produce that type of immobilized enzyme. So our interest in this particular area came from our studies on a protein in Bacillus thuringiensis called cry protein. And what's novel about these cry proteins is they form these crystalline inclusions like this. You can isolate these in crystals. And actually, people have taken these crystals and done X-ray diffraction studies and shown they are actually true crystals. So they would basically take the cell, send it through a femtosecond laser, X-ray laser, and look at the diffraction from those crystals. And from a whole series of those pictures, they can solve the structure. So what these crystals are used for normally, and they're actually very important, is that the bacteria grow on plants. And what happens when insects, such as this worm, eat the crystal, it dissolves in the gut of the insect and basically forms a activated toxin that inserts into the cells of the insect and kills the cells. Okay, and so it's actually used in the biotech industry by Monsanto and Dow to make genetically modified crops that are resistant to insects. Okay, and so these crystals are important. They're not toxic, and also they actually turn out to be very stable. They actually can make like cakes of these bacillus, store them, and if they want to treat a pond to remove it of mosquitoes, they dump the cake in the pond, and then the crystals are released and they kill the mosquitoes in the pond. So it's a very stable platform. Here's the structure. Since I'm a crystallographer, I'll show you a structure at least that was solved in 1991 by Jade Lee et al. And basically the crystal consists of an alpha helical domain and two beta sheet type basically containing domains. Okay, so our interest initially was actually to do some crystallographic studies of these, and we were trying to insert them into membranes. And to help in characterizing whether they were inserting or not, we decided to fuse a GFP or M-cherry protein to the C terminus of Cry3A, which is the protein we're working on. And as you can see, when we do that, the Bacillus thuringiensis cells are either green if we fused a GFP, or red if you fuse an M-cherry. We didn't think we planned on isolating the protein and solubilizing it. We didn't think they'd form crystals, but to our surprise, when we harvested the cells and we did a density gradient centrifugation, here's the centrifugation, you could get a layer shown here, which turned out to be pure crystals. They had the full-length fusion between either Cry3A GFP or Cry3A M-cherry. And we could actually isolate these crystals as shown here. So here's Cry3A GFP. You can see all these little dots. These are isolated from the cells. They have really nice shapes. And a little lighter color, you can see the M-cherry crystals shown here. And there's two important things. First, that the crystals are consistently about the same size. And the other important thing is that the protein that's being fused to the Cry3A is functional. And we've also done this with luciferase as well. Here's an EM of the crystals. You can see that both the Cry3A GFP and the original Cry3A crystals are about the same size and look about the same. So in the intervening years, we've developed several different technologies to anchor enzymes to Cry3A. One is a fusion technology. Here we take the Cry3A gene, we fuse the enzyme of interest to make a Cry3A enzyme fusion, and we isolate the Cry3A enzyme fusion crystals. Another system is we use SpyCatcher SpyTag technology, and we take Cry3A crystals fused to SpyCatcher, and then we subsequently add the enzyme of interest with a SpyTag, mix the two, and the enzyme will add to the SpyCatcher, sorry, and we get our Cry3A SpyCatcher SpyTag enzyme fusion. But perhaps the most interesting one is the cryo-entrapment technology, which we've discovered most recently. And here what we do is we co-express the Cry3A protein with the enzyme of interest, in this case it will be PML, in the bacillus cell. And so it'll form this crystalline lattice. The Cry3A proteins will form that lattice. But it turns out with the right protein, the target enzyme will fill the channels of the Cry3A crystal, because Cry3A has these really nice channels. And you can actually, it'll be entrapped, they don't leave. And then you can add substrate to it and they'll convert the substrate to product. In the entrapped protein will convert the substrate to product. So our initial system that we got to work were with lipases. We focused on that because they're actually the most common industrial enzyme used for biocatalysis. They're used to produce biodiesel, they're used to produce cosmetics, they're used to produce pharmaceuticals. And they estimate the global market was in 2020 about $590 million. So it's actually a big enzyme market. Okay, so we tried different technologies. We tried a fusion technology and we tried the entrapment technology. And here you can see that the entrapment technology, which we use with brackets, is about 10 times more active than the fusion technology. So the type of way that we immobilize the enzyme with our cryo-3A can make a difference. But the key advantage, at least for the system, is its ability to enhance the stability of the enzyme. So here's the free PML protein, and in blue, and in red is the PML entrapped in cryo-3A. And you can see at 55 degrees, the free PML protein is dead. But for our cryo-3A PML entrapped, PML trapped in cryo-3A, you still have about 60% activity. And so we think this is because when you entrap the protein within the crystal, you prevent the protein from unfolding, and thus you're able to enhance its stability. And that's important for industrial catalysis because you can raise the temperature. So we then used it for a application. So one of the applications of lipases is, and particularly PML is good for this, is to take unrefined waste cooking oil that you go to like Maxim down in SHO. They produce a lot of waste cooking oil. You can take that waste cooking oil and turn it to something useful, biodiesel. So basically you take the lipids that are in the cooking oil, you add PML, you add some methanol, and then it'll convert it to glycerol and FAME, which is the biodiesel that's used. So basically mix the waste cooking oil with the PML, you get the biodiesel. And what's nice about the crystal is that you can recycle it. So here we go through 15 cycles, and you can see we're able to retain most of the activity over time. And I think most of this loss of activity is the loss of crystal in the emulsion between the oil and water layer. And if we can stop that loss, we can probably maintain that conversion much longer, and we're working on it. So how does this technology compare to the existing immobilized lipases? Here's the relative activity. You can see that our cryo-fusion and entrapment technologies are better than any of the existing gold standards, particularly Novozyme 435, which is the best system right now. Ours have higher activity per unit mass compared to that system. So we are competitive with that system. Okay, so after having utilized this technology for hydrolytic chemistry, we decided we wanted to try to use it for redox reactions. And so the QIN announcement was made, and we found a partner, Ying Wang, who was working on electrochemistry that could help us to try to expand our cryo-3A technology to redox enzymes. And here's Ying. And the team that we assembled over the time are shown here. Xu and Wen both started the project, and later Reza and Yekta and Zanyu started a second project, which due to time limitations, I'm not going to be able to talk about today. So the system we decided to focus on for one of the studies was the P450 OLET fatty acid decarboxylases. These take fatty acids, so it's kind of related to our previous work, and hydrogen peroxide, and they turn them into alpha olefins. So basically they decarboxylate, take off the CO2 and make a double bond here. Or they can hydroxylate it, okay? But this is the one we're more interested in. And these alpha olefins are used to be a biofuel itself, but they also can be precursors for other downstream uses, like lubricants. So the first step was, this is all Ying's work I'll talk about for the next several slides, was to make a electrode that could produce the hydrogen peroxide in a continuous fashion so that we could eventually use it for our enzyme system. And so for this study, she developed a three-phase flow cell system, which basically consists of a gas diffusion electrode. So the O2 is going to come in here, and it's going to be converted from to H2O2 at that electrode. And then you have you have the counter electrode on this side, and you have a mesh here to block the flow of protons. Here's a picture of that electrode shown here, and basically it just shows you the size of the the electrode system. So then she had to make the electrode that's going to be used to make the H2O2. They developed a method to make a cobalt mesoporous carbon catalyst. They may take 1,10-phenanthroline, make a cobalt complex of it, and then they pyrolyze that cobalt complex in the presence of glucose, and then do with some grinding and acid wash to make a porous carbon surface with some cobalt sites. And here's an SEM of that picture of that catalyst. This catalyst has a surface area of about 392 meters cubed per gram, and it has an average pore width of about 5.285 nanometers, meaning that the oxygen can get in and out of that electrode. The nice thing is the H2O2 selectivity. Here you can see it's primarily making H2O2, and then over time you'll notice that the H2O2 concentration increases in a weight percent in solution as the catalyst is working. So this allows you to do continuous production of H2O2 using this oxidation O2 reduction reaction. So the catalyst is ready, and now we have to work on trying to anchor the crystals with the enzyme onto the surface. So the way that Wenbo and Xu focused on was to develop take the electrode and try to anchor it with an ionomer, which is called naphion. And so what we want to do is have the crystals go onto the surface and be kind of held down by the naphion or ionomer on that surface. Here's the naphion and here's our crystal. So here's the apparatus used to do that. Here's the carbon paper that they're going to use to make the electrode. They spray the Cry3. For these studies, we used M-cherry crystals because they're fluorescent. We sprayed Cry3 M-cherry crystals and the ionomer onto this surface to get an even film. And then we tried to look for whether the crystals would stick. So here's a study done by Xu, and I show this picture because I found Xu's solution to the problem kind of clever. So he needs to figure out, he needs to get the fluorescence off of this carbon paper or fiber. So the way he got the fluorescence is he used a punch hole, like the paper punch hole, and then put those paper punch holes into a 96 well plate and measured the fluorescence of the paper in the 96 well plate. And it was able to get the fluorescence out of that. So I thought that was very clever. So anyway, the carbon fiber itself has no fluorescence. If you add the Cry3A crystals, you can see there's fluorescence as you expect. But now, this is after 3 days air drying. Now you wash the crystals, and if it's working, it should stick. And so he's able to wash it and only lose about 60% of the fluorescence. Thus, we're able to get the crystals to stick to the surface. And now we can start to try to do the OLET. So we tried 2 different technologies to get the OLET, Cry3 OLET crystals. OLET is big, so we didn't try entrapment, but we did try fusion. And we also tried our Cry3A SpyTag technology, where we had the OLET come in. We made the OLET separately and then coupled it. Okay, so here's the mass GCMS result for the OLET SpyTag. You can see after incubating with OLET SpyTag, we incubate lauric acid, we decarboxylate it, it will become undecane. Here's the standard. And you can see we get quite a bit of undecane, more than if it's not definitely linked, but you can see we get a lot of undecane, undecene, sorry, enough that we can detect it in good amounts. Xu then tried to do the coupling reaction of OLET SpyTag to Cry3A SpyCatcher. Here's the OLET SpyTag protein. Here's the Cry3A SpyCatcher. And then you can see when he mixed the two, he can get an extra band that's the combination. So he's able to get the anchoring of the protein. The unfortunate thing at this stage is that the conjugated products show no activity. So at least this route to producing the OLET in an active form isn't going to work. So we also looked at the fusion, and it does have activity. We can see the undesine, but unfortunately, it's not very high. So we're working to improve the activity. We have some strategies to do that, and I think we'll try to solve it over the next year. So at least I don't, in the interest of time, I only talk about one of the two projects. The other project is on using formate dehydrogenase to make CO2, but I'll keep that out for this story. So just to acknowledge the people that did the work, Xu in my lab and Wenbo Xu in Professor Ying Wang's lab, this is a collaboration, worked on this OLET project that I talked about today. Reza Inyekta and Zanyu Xu are working on the second project that I didn't have time for. Marianne, Brad, Manoj, and Tian did the other Cry3 work that on the lipase. And we want to acknowledge, of course, the people that funded. We thank the Faculty of Science for the CRIMS funding, CUHK for the PIF, which I guess is a 50-50. We also have gotten RGC funding for the lipase work and ITC work for the ITF work as well. So that's the end of my talk, and I'd be happy to answer any questions. Thank you.
Okay, thank you, Michael. I'd like to, any questions from the floor?
Yes, Michael, thank you for an interesting presentation. I never learned how the enzyme actually worked until hearing your talk today, but some of the specific system also relevant to energy. For example, in the biodiesel production cases. Now, in the hydrolysis state, I think your PML is catalyzing the hydrolysis in the ester bond. And then after
it's a transesterification, yeah, but it's a transesterification, not really.
Yeah, it's transesterification, but after the production of the biodiesel, you actually end up with carbonic acid and and the cholesterol.
No, we end up with an ester. It's an ester.
You produce glycerol, right?
Yes, glycerol. You basically you add you add the methanol to here, and then you basically lose the glycerol, and you have a methyl ester of the fatty acid.
Okay, I see. You are producing methyl ester. Well, I see that with the carboxyl group and also the trihydroxy group in the glycerol, wouldn't that actually bond to your PML, causing to lose them gradually, rather than just a oil-water interface? Because this is a strongly polar and with a strong hydrogen bonding system.
We don't think so, because we can see that right now we can see particles of our crystal kind of at that interface. So it's possible that those are involved, and we have a solution that we just submitted a grant to try to overcome that. But I think that's more the issue. That I don't think it's the glycerol, because the glycerol is pretty soluble in I think it's in the water layer. I think that's where it goes.
It is a fully soluble in water, and that's why
so
I saw that the interaction with hydrogen bonding, which is, by the way, often the cause in the loss in the catalyst in the recycle systems.
We can take a look at that. That's a good suggestion. We actually have a way to convert the glycerol to alpha GG. Maybe if that's an issue, when we convert it to alpha GG, then we can overcome it.
Yeah, that will substantially reduce the bonding interaction.
Okay, we'll keep an eye on that if we get the alpha GG conversion to work. Thank you.
Thank you.
Okay, thanks, Chunshan, for the question. Any other questions? Maybe I have one actually, Michael.
Okay.
You mentioned, because I guess you know the sort of the crystal packing. I think it's a brilliant idea to trap the enzyme in there. But can you estimate the size limit based on the structural information that how large the enzyme can fit in the, the gap?
Yeah, in our JAX paper, we actually give the numbers. I don't have, I'm sorry, I can't remember the numbers on the top of my head, but we actually have calculated those numbers. There's a program, MAT Channels, that will actually show you that. We estimate it's typically, of course, the dimensions of the protein matter, but typically about a 20 to 30 kilodalton protein will fit. It has to be big enough that it will not get, it can't get out of the crystal, but it has to, but it can't be too big because it has to fit in the remaining hole. But we do have that estimate in the paper.
Okay, thank you. Okay.
Can I ask a quick question?
Sure, Li Wen.
Michael, nice talk. I think you made a GFP M-cherry fusion. Looks to me the GFP, the punctures, more uniform than the M-cherry, right? Are they the same, identical? What if you make an N-terminal fusion? Would they be the same?
What do you mean? Sorry.
Did you look at this picture, right? The green force and the puncture, look quite uniform. The size also slightly different from the M-cherry one.
We haven't. Part of it is that somehow GFP shows up nicer on, I don't know why, but they should be similar. GFP and M-cherry are the same, are, just mutants of one another. Right. So it's possible, but that they're different, but I wouldn't expect them to be too different. I think it's just because the GFP shows up brighter than the M-cherry.
Did you also try to make an N-terminal fusion?
N-terminal fusion will not work for Cry3A because the N-terminus gets cleaved quite often. It's just for some reason, it just naturally self-cleaves. Okay. So there's another one, Cry1AB, that will, N-terminus works and the C-terminus doesn't work. So it just depends on the Cry protein.
Okay, thank you.
Thank you. Okay, I guess we are a little bit, a little bit overrun, and let's thank Michael for his excellent talk again.
Thank you.
Thank you. I'll pass the time back to the MC.
All right, thank you, Professor Wong, and thank you, Professor Chen. So we'll now move to the next presentation. May I now invite Professor Jianfang Wang, the chairperson of Department of Physics, to introduce our next speaker, Professor Lu Xinhui. Professor Wang, please.
Okay, good morning, everyone. I'm very pleased to introduce our next speaker, Professor Lu Xinhui. Professor Lu obtained her bachelor's degree from Nanjing University in 2004 and a PhD degree from Yale University in 2010. She then worked as a postdoc researcher at Brookhaven National Laboratory before joining our department in 2012. She was promoted to associate professor last year. So her research interest lies in energy-related materials, also soft condensed matter physics, so including organic and perovskite solar cells and functional thin films. She is an expert on X-ray and neutron scattering. So she is now serving as the council member of Physical Society of Hong Kong and the Chinese Neutron Scattering Society. So Professor Lu has been extremely productive in research in the recent years. According to a Google Scholar, she has published close to 300 papers with an H index 65. So she will talk about thin film solar cell studies with grazing incidence X-ray and neutron scattering. Professor Lu, please.
Thank you for the kind introduction. Let me share my screen. Okay, I'd like to thank our faculty to give me this opportunity to share our group's recent progress on third generation thin film solar cells with grazing incidence X-ray and neutron scattering techniques. So our groups are focusing on studying the organic and perovskite solar cell. Both of them are belong to the third generation thin film solar cell. So both of them have attracted extensive research efforts, and their efficiency are for organic solar cell is approaching 20%, and perovskite solar cells over 25% nowadays. So our group's research interest is in understanding the multi-length scale microstructures in the active layer, the light absorbing layer in the solar cell. So for organic solar cells, the organic donor and acceptor materials, they mix together to form this active layer so that we can have abundant interface for the external dissociation. So here we are interested to see how does this organic molecule pack in the semi-crystalline domain, and which is the atomic scale microstructure, and how what is the crystallinity and orientation will affect the charge transport mobility. And how does this donor and acceptor nanophase separate into different domains, which will affect the external dissociation and charge collection process. This belongs to the nanometer scale structure. And for perovskite solar cell, the crystal structure are well defined. Basically, this is this, so all of them have this ABX3 chemical structure, and the BX6 octahedral share the corner and form this cubic cage, and then the A site large cation insert into this cage to stabilize this cubic structure. And depending on the ionic radius of ABX, the crystal structure can be cubic or distorted a little bit, become tetragonal phase, we call beta phase, or distort further, become orthorhombic, alpha phase. All these corner sharing phase are black phase. And if there get further distortion, it can be collapsed into unwanted yellow phases. We call it delta phase. So these are atomic level or angstrom level microstructure we're interested in. And then going to a little bit larger scale, if the grain size in the bulk film, or if the active layer are formed by quantum dot, perovskite quantum dot, how does the quantum dot pack? These kinds of nanometer scale structure will also affect the device performance. So to probe this kind of microstructure, we are using the grazing incidence scattering. So basically, we are making use of this scattering wave vector Q as a prober to probe the 3D order structure inside this thin film. So this the Q should be aligned into the normal direction of the order structure. Then we can probe that structure. So once our incident beam and detector are fixed, so this Q is fixed. So to probe different orientation of the structure, we have to rotate our sample into this reflection geometry so that this Q can have both QZ and QR component to understand the out of plane and in plane structure inside the thin film. Then to understand the multi-length scale structure, we basically just move our detector when the sample to detect the distance. When it is close to the sample, we are collecting wide angle scattering, which corresponds to small feature, angstrom levels feature. We call this GWAX. And if we move our detector far away from our sample, we are collecting small angle scattering, which is correspond to larger scale, nanometer scale structure. Okay. So here, let me give you some examples of how we use GWAX to understand the molecular packing structure inside the organic solar cells. So the traditional, the old generation organic solar cells are made of this P3T and PCBM, and P3T will pack into semi-crystalline structure. And when we see this GWAX pattern, and these peaks are lamellar peaks, it appear at small q in the QZ direction. So it means that these are relatively large lattice constant and in the out of plane direction. So lamellar direction in the out of plane. And then this peak appear at relatively larger Q, and in the QR direction, this corresponds to the pi-pi interaction in the in-plane direction. And then we study the packing structure of the milestone family, ITIC family, in collaboration with the leading scientist, Zhang Xiaowei, who is the inventor of this family. And we find that for this molecule, the pi-pi peak going to QZ axis and the lamellar peak going to the QR axis. So, which means this orientation of the molecular packing rotated 90 degrees become face-on packing. And on this QR axis, we observe another set of periodic bragg peaks, means that there's a new structural order in the in-plane direction. So indicate that these molecules, okay, with this rigid linear backbone, tends to have this N-group pi-pi stacking, which will form a new structural order, backbone order, to facilitate the electron transport in the in-plane direction. So then we go into the next generation, this Y6. So this is another milestone molecule synthesized by Zhou Yingping in Central South University, and which pushed the efficiency of organic cell over 15% in recent years. So compared to the ITIC family, Y6 has this L-shaped backbone. So whether this Y6 can also have backbone order in the in-plane direction. Okay, so Y6, the GWAX pattern, the crystallinity is much lower, so we cannot guess what is the packing structure just by these blur peaks. But we see two blur peaks, so which indicates that there might also be some additional structural order in the in-plane direction. So to understand the backbone packing in the in-plane direction, we heat the sample to promote the crystallization. So we can see when we increase the temperature, the bragg peak become more and more clear, and at 250 degrees Celsius, the pattern switched from the pi-pi go to the QR axis, and the in-plane folded bragg peak that is folded in the in-plane direction become rotated 90 degrees, so that become this discrete square lattice pattern, so that we can understand this geometry. So it's like it rotate from edge on face on to edge on, so that now we can index this bragg peak. And we find that Y6 have this two-dimensional backbone order, and this will facilitate the electron transport in its in-plane direction and find its connected pathway to the corresponding electrode. So then we try to use small angle scattering to understand the 3D nanogeometry, okay, nano scale morphology, to understand how does the donor and acceptor phase separate in the active layer. So here in convention, so we are using GSEX, which is collecting the scattering signal above the surface horizon of the sample. However, so this technique theoretically can also get the vertical nano morphology, but there is the surface enhancement peak called the unit peak that shadows the vertical structure here. So we are trying to making use of the scattering signal that escape from the front edge of the sample, which is called the grazing incidence, but transmission signal, to get this get rid of this unit peak and get the clear vertical nano structure. So to get such kind of signal called the GT-SEC signal, we have to cut our sample at the center, make a clean edge like we create for the cross-sectional SEM measurement, and then we place our sample at the center of the rotational stage, shine our incident X-ray at the front edge. So in conventional setup, the X-ray is shine in the center of the film, so the X-ray signal cannot escape from the substrate, okay, because the substrate is very thick. But if we shine this X-ray at the front edge, then the pathway for the scattered X-ray can escape from the front edge, and then we can get below horizon signal. So by this way, we can get the direct comparison of G sex and GT sex in the same scattering pattern. So this line just separates the surface horizon, and when we gradually increase the incident angle, we can see that this unit peak was lifted off, and we can get symmetric scattering pattern below horizon. And by fitting the scattering pattern both in plane and out of plane, so in G sex, the in plane domain separation feature we can extract as convention, and by GT sex we can get similar structure. Okay, and in the out of plane direction, this peak is from the unit peak, which is unwanted. But for GT sex, now we can also get clean scattering feature from our domain size. So by this, we can, for the first time, we can extract the out of plane nanophase separation information of the sample. So we use this to study the classical DBT ITIC system. Find that so inside this organic active layer, the domain, so the vertical, so the domains are not isotropic. So in the vertical direction, the phase separation domain size is shorter than the in-plane direction. So this will become the bottleneck for the external dissociation. Okay, so we can understand the device behavior in a new window, basically, to understand it in the vertical direction because it's shorter. Okay, and due to the time limitation, I think I just skipped this slide. So basically, we learn now we have this handle, and so we can tune the vertical morphology and to get a very good device. And for the Prof's guide solar cell, since the crystal structure is very clear, so we're trying to use in-situ study to understand the phase transition pathway during the film formation process by mimicking this kind of fabrication process at the synchrotron beam line. Okay, so we mimic one-step and two-step method and trying to see what is the difference of different recipe. So in this work, we trying to understand the during the one-step perovskite fabrication, when we dope the A site cation with doping cesium or without cesium or with cesium, what is the crystallization pathway? So without cesium, it undergoes 3 stages, and the stage 2 is the annealing window. Stage 2 is very short. So our device, if the anneal outside this annealing window, the device performance will decrease significantly. But when with cesium, the stage 2 is elongated quite a lot, and then the reproducibility of our device increase significantly. Then we try to understand the 2-step fabrication technique. So for this, we can do A-site doping both in the first step and in the second step. Okay, so in the first step, if without cesium, it started with lead iodide, but with cesium, it forms some small scarce nucleation center of delta cesium lead iodide phase. So the formation of perovskite are preferred to start with this delta phase. So this will significantly increase the grain size. However, in the second step, if without the doping of GA, the large cation, then the crystallization speed is relatively slow, which will lead to pinhole on the film. So this is no good. So then with the doping, we can promote the crystallization in the second step and form this pinhole-free and larger grain size process cell, which lead to a very good device. So recently, we trying to understand the phase transition of perovskite quantum dots and how does the PAC affect the phase transition. So the behavior is quite different from the bulk film, because the surface ligand on top of the quantum dot will stabilize the black phase, which is not capable in the bulk film. However, when we heat the sample, it will still transfer from black phase to yellow phase. But if we slowly heat the sample, it can stabilize in the black phase. We find this is related to the packing, the heat during the packing. The quantum dot tends to pack in a more ordered structure. This will increase the lattice strain inside the perovskite quantum dot, and which suppress this unwanted transition. So then in the following work, we try to pack the film, the quantum dot film, as dense as possible by mixing two different sides. And then we find that indeed in the mixing binary mixing film, the packing density, the volume fraction of the packing density increase, which leads to the increase of the device performance. And we are in collaboration with Professor Wang Yi in our department to understand why the binary quantum dot can pack better, okay, which is intuitively depending on, because we are having two different sides, so then the small quantum dot can insert into the gap in the large quantum dots packing. However, I think so here it's more complicated than this, because the ligand density will also affect the packing. And we also have some efforts in the to understand how does the intracranial how does the operation condition of the perovskite solar cell, and we find that for inorganic perovskite solar cell there is a significant light soaking effect. And so with our technique, we try to shine our sunlight and to understand the lattice changing, and how does the defect change the passivation condition with respect to this working condition, and to passivate this, to suppress this kind of or expedite this light soaking effect. Okay, so for the last part, we recently trying to establish this grazing incident scattering technique at the Dongguan neutron spallation neutron source. And so basically, because for X-ray, their scattering contrasts are depending on the electron density of the elements. So it is not very sensitive for organic materials. But for neutrons, it interacts with the nuclei of the sample, and the scattering cross-section is irregular. So for X-ray, basically the contrasts are mainly originate from the contrast between the crystalline domain and amorphous domain. But now we're trying to use neutron to deuterate our sample selectively, so that it can tune up the contrast between donor and acceptor, and so that we can have understanding on the phase separation in the amorphous region. So our preliminary result tells us that the deuterated the non-fluorine acceptable Y6 has about 3 times of the scattering length density of the undetected molecule, and which will give us significant contrast between this donor and acceptor. And yeah, so we can have a better understanding of the morphology inside organic fields. Okay, so with this, I would like to summarize that our research always rely on collaborations with expertise in material synthesis, computational simulation, device engineering, and device physics. So we are always open for collaboration. And recently, we're trying to build a team to shoot for a collaborative research project for flexible perovskite solar cell with scalable techniques. And we are also open to any collaboration for any interesting system and phenomena. If it needs our understand the 3D multi-lens scale structure of your sample, feel free to contact me. Okay, and I'd like to thank for the group member and our collaborators, Bing Time from Dongguan Neutron Source and Taiwan Light Source and Shanghai Light Source, and the funding support, especially the seed money from our Faculty of Science to stimulate our collaborative research. Thank you.
Okay, we thank Professor Lu for the talk. So now we take a few questions.
Could I ask a question?
Yes, please, Dean.
Okay, Xing Hui, excellent presentation. Thank you very much. This is very interesting. I noticed that when you did the backbone structure analysis, I don't remember whether you used the GSEX or not. And when you heat it up, the temperature and then the structure order changed dramatically in a way. I was wondering that when you decrease the temperature again, will the structure relax and come back or become sort of locked in the new structure at the elevated temperature?
Very good question. So here, I think we haven't reached the melting point of that material. So if we heat up the sample again, the crystal structure will remain there unless we heat it further to reach the melting point, and then actually will break down the molecule and lose its crystal structure.
I think my sort of chemistry mind began to wondering whether that the sp3 carbon rotate to find a more comfortable structure when you heat it up, which does not happen because they're structurally sort of fixed at a lower temperature due to the structural rigidness. But when you heat it up, sp3 carbon become able to rotate, the structurally more relaxed, and then help to create a better intermolecular interaction in a different sense. I don't know whether that.
I think so. So basically, for our technique, we don't know the details about these functional groups. So we can only know their structural order. So we have to in collaborate with computational simulation to understand. So for this side, actually, if we can grow single crystal of the sample, so then we can understand. But then the other question is whether this single crystal structure exists in the thin film, because in the thin film, the structure is not that well ordered. So it's relatively disordered structure. Yes.
Yes. A related question on this. When this improves the solar cell performance in the organic solar cell, does it enhance light absorption, or does it stabilize the charge so that the photoelectrons and the holes are better separated?
So basically, this is for the charge transport. So it needs to, for the charge transport, because it needs to form this kind of overlapping electron cloud to let the electron transport through the backbone and the pi-pi interaction. So if we see this kind of backbone structure, means that there is overlapping in this pi-pi functional group, and then this will form a connected charge transport pathway so that after the external dissociated, the charge can transport to the corresponding electrode.
So this type of pi-pi related structure or the aromatic cross-linked structure better stabilize the photoelectron or better stabilize the holes?
It's not to stabilize the electron, it's for the charge transport, basically.
Make them move more mobile?
So make it the mobility more, so the charge carrier mobility will be higher. Yes.
Oh, I see. Excellent. Thank you. Very interesting.
I have one question here. So as I can see, is diffraction forms rings. So some rings are very sharp with a small thickness, but some other rings they are very thick. So can you also extract some information about the size of the the domains of the grains?
Yes. So basically, the width of the peak in different direction tells us the scales, the length scale of the structural order in certain direction. So for example, if this kind of banana shape peak, so in the QZ axis, if we do the line cut, it will tells us the correlation length, how the distance for this kind of structure to lose its coherence. Okay, but if we do a line cut in this angle axis, it means the orientation of this kind of structural order. So if we span a longer angle, it means the orientational order is not good. Yeah, we can also extract this. So this is in the reciprocal space, everything is in reciprocal relationship with the real space structure. So if it's wider, means it's short, the structural order is shorter. So if we concentrate it in the discrete peak, means it's kind of single crystal structure.
Okay, any other questions? So if not, because we are behind the schedule, so we stop here. We thank Professor Lu again for the interesting talk.
Thank you, Professor Wang and Professor Lu. So we will now have a 5-minute Zoom break, and our next talk will start at 11:33. Please stay connected with the webinar. Thank you very much.